大家好，我係今日講座嘅司儀 Natalie， 歡迎大家出席由香港科學館、香港中文大學新亞書院同生命科學學院聯合主辦嘅任國榮先生生命科學講座。今次講座，我哋有幸邀請到德國康斯坦茲大學水生系統適應遺傳學教授、國際珊瑚學會主席 Professor Voolstra 為大家主講，題目係珊瑚礁與氣候變遷、緩解生態崩潰。我哋亦邀請到香港中文大學李福善海洋科學研究中心主任羅海偉教授主持講座。除咗喺香港太空館嘅現場講座會同時於 Zoom、香港科學館 Facebook 專頁同 YouTube 頻道現場直播。講座完結後設有問答環節，如果大家有任何問題，歡迎屆時舉手發問。收睇網上直播嘅觀眾亦可留言發問，我哋會因應時間代為向 Professor Voolstra 發問。喺正式開始之前，我哋先邀請香港中文大學理學院及生命科學學院院長陳浩然教授為我哋簡介任國榮先生生命科學講座。有請陳教授。
So, Professor Voolstra, Professor Hector Chen, head of New Asia College, and all the college members of New Asia College, and also my colleagues and friends and students from the Chinese University of Hong Kong. I'm so glad to be here today in my capacity as a director of School of Life Sciences, Chinese University of Hong Kong, to introduce you the lecture. So as you can see here, this is the Yang Kuo-Yung lecture in life sciences, and this is also the 10th lecture since we started this lecture series. So I'm so grateful to here to explain to you about the history of this lecture series, which was started in 2015 with the generous support and donation of a New Asia College from a family of Dr. Yang Guorong, founding chairman of the Department of Biology of the college. And each year, we have invited different distinguished scholars, biologists, and more recently, we all call ourselves life scientists to share with us their views on certain aspects of life sciences. And the lecture series actually have broadened the knowledge of life sciences among staff, students, and as of today, the general public. And today is the third and also the final lecture of Professor Voolstra. And Professor Voolstra actually already given a lecture at the New Asia College Assembly, a research seminar at the School of Life Sciences, and today he will be sharing with us a topic on coral reefs and climate change, mitigating the ecological collapse. So I have attended the previous two lectures. I'm very, very looking forward to Professor Voolstra's today's lecture, which would actually bring scientific investigation, life sciences knowledge, and the conservation concept together. And I hope you all enjoy the lecture. And I also encourage you all to participate in the question and answer sections, and we will have a very enriched discussion and interaction. Thank you very much.
Hello, everyone. So my name is Haiwei Luo. I'm from Chinese University of Hong Kong. So I do marine microbiology and coral microbiology stuff. So today it's my great honor to have our distinguished professor, Professor Voolstra, Chris Voolstra, to visit CUHK and to deliver one of the talks that is facing to the general public. So let me briefly introduce Professor Voolstra's biography. So he received undergraduate degree and PhD degree from University of Cologne, Germany. So then he started actually his own career at KAUST. KAUST is King Abdullah University of Science and Technology in Saudi, which is very famous for coral research. Then Professor Voolstra further advanced to full professor and accepted a very distinguished position at University of Constance, Germany. So he is dedicated in genetics of adaptation in aquatic systems. So Professor Voolstra holds many distinguished positions. Among these, I'm not going to through this whole very long list, okay? But I would just say he is current our society chair, president. That is the society. Our society is the International Coral Reef Society. Okay, so he's currently the president. So he is super productive academically. He's currently already published over 300 peer-reviewed publications and got almost 30,000 citations. For people who know science, so we all care about H index. So he got H index more than 90. So this is very rare for marine ecology scientists. Okay, so among these, so he published many top-tier journals. So without further ado, let me invite Professor Voolstra to your stage.
I'm going to try to do this while I stand. Otherwise you won't see me, and I also need to move around. It's easier for me. Thank you so much for inviting me. I really had a great time so far in Hong Kong. And Highway, one reason why I have so many papers is because of colleagues like you and collaborations. This is really the essence of research, like working together on problems. I also want to point out that the first speaker is actually here from the lecture series. So sorry, I forgot your name, but it's a great honor to have you here and to actually make the effort for you to come. So of course, pressure is on me now to make it worth your time and all of your time. I'm gonna talk about coral reefs. Can I quickly ask who's actually a student? Okay, students, just, okay, good. You will see in a second why, but we can already jump right into the topic. So what you see in the background, that's actually a little boat, which we call a dinghy, and these dark things underwater, this is actually coral, right? So it's a huge organism. It's actually the biggest biogenic structure we have on this planet. But very briefly, and this is pretty much why I asked who's a student, if I would sum up my career in one sentence, it was completely not planned, okay? I started with fruit flies, I changed to mice, was still not super happy, then I went for a postdoc to Mexico to do field work, and then I traveled to Saudi Arabia. As you can imagine, my parents were pretty excited. But this kind of all the time I followed my heart, and I think I found what I really like and what I'm good at. And if you're a student, I also encourage you to do this. Like, it's not a problem to transcend different things. Throughout my career, I did very different things, but every time I went from one place to another, I brought something to this place which people didn't have, and I learned something from people there which I don't have. So it's it always looked like a bug, but it's actually a feature. So it helped me a lot in my career doing this. Okay, and of course the irony is now I live at one of the biggest lakes in Europe doing marine biology. So yeah, you do the math. Okay, but let's jump right in. So I already said it, coral reefs are the largest biogenic structures on Earth. So what we look at here is actually a picture taken from the moon by NASA. This is the Great Barrier Reef in Australia. It's the biggest coral reef system in the world. But I mean, there's hardly any other biological thing that you can take a picture from the moon and you can still recognize it, right? If you zoom in a little bit closer, this is actually what you see. And Charles Darwin, the founder of the theory of evolution, he was actually also really interested in coral reefs. And he called it the oasis in the desert of the ocean. Which is for two reasons. A, because coral reefs are actually incredibly rare if you consider the entire size of the ocean, so only 0.1 to 0.2% of the ocean surface are coral reefs. And B, because you can see this very nicely here. You have this crystal clear blue water in the background, and only where you have corals, you have all the life. So coral literally are the oasis for all the life in these ecosystems. And then if you further zoom in, which is also, I think, a very nice picture to illustrate, right? Like anybody can easily recognize 20 different species here. And coral reefs are actually the rainforests, or termed the rainforests of the ocean. So 25% of all described marine species are tied to the presence of coral reefs at one point in their life. And they provide goods and services for people. So you're probably more aware of here because you have the ocean all around you. And of course, fish is a very important source of protein. Then this is a picture from Egypt, tourism. This is actually a benign picture. I've been there 10 years ago. Every day in the year, there's 100 boats of this size going to a small coral reef, and they deploy 1,000 divers. And these corals are still there. So we have to acknowledge that corals are sensitive to environmental change, but on another note, they are also very resilient, right? If you think about that, somebody, 1,000 people come to your house each day and have a look around, and you could see how your house looks like after a year, right? And then they also, coral reefs also provide shoreline protection, which you can see here in the lower right. This is actually a huge barrier, and people calculated the wave energy that coral reefs absorb, and it equals basically the energy of a nuclear bomb in many places. So there is a lot of energy from the ocean that the coral reef takes away, and therefore they make the shoreline more stable. So these are all good things, but and this is all pictures I took from my various trips. So of course there's local problems, right? Like we produce a lot of garbage, or we have produced a lot of garbage. I'm actually, I have to say, I rarely came across plastic waste here, which is very good that you have such a good effort in Hong Kong of avoiding this. But this is how the shore in front of a coral reef looks like in one of my expeditions in the Coral Triangle. Then, of course, the major big cities of this planet historically have been either built at large near rivers or the ocean, because it's very easy to get rid of your waste, right? And the last local threat, so below this is, I have to look at Palm Jumeirah. So this is an artificial city in Dubai, which it's a bit of a sensitive issue, but there were coral reefs there, and it's not exactly clear whether they used coral reefs as a foundation for this, but it just highlights this bigger issue that everybody wants to build close to the ocean for various reasons, and of course this affects the life in the ocean. You also have a lot of shoreline construction here, right? And it's important to figure out that we do this sustainable and in harmony with nature. But besides these local threats, there is one global threat, which is climate change. And I was also very pleasantly surprised that this is very much acknowledged here, right? I heard that people that live longer in Hong Kong, they actually experience the change, that you have more hurricanes or tropical storms, so they are more often and they are more severe. And I think this is a very good graphic, because it just conveys the numbers that we are looking at. So what is basically plotted here is the atmospheric CO2 concentration in blue against the man-made carbon emissions in gray, right? There's almost a one-to-one correspondence. So I think you don't have to be a scientist or a researcher to see that there's a very high likelihood that one basically makes consequence of the other. And then plotted over this is the temperature anomaly. Yeah, this is pretty much a global threat for corals, and I'm telling you in this slide why. So we all have probably heard climate change or ocean warming, but a little acknowledged fact is that 93% of this extra heat energy actually go into the ocean, okay? So the ocean is the biggest buffer of warming that we have, and without our oceans, it would probably already be very unbearable to live here on this planet for humans. So closer to my home turf or my scientific expertise, what this ocean warming does, it triggers so-called coral bleaching. So what you see here in the middle is a coral colony, which is this spooky white. Okay, so bleaching describes this physical process of whitening. It's important to acknowledge that these corals are not dead. Okay, they are basically negatively affected, and what actually happens in bleaching is depicted here. So a lot of times people describe corals as colorful rocks. Okay, they are not rocks. They are also no plants. They are animals. They are sessile animals, so they attach to a place and then they grow from there. But they do one specific thing that only a couple of animals do. They actually associate with tiny plant cells, which we call algae, and these tiny plant cells live in their tissue, and you all know that plants do photosynthesis, right? So these algae do nothing else. They do photosynthesis, and photosynthesis is basically converting sunlight energy into biological energy. So in the process of doing that, they create sugars and other nutrients, and these they give to the coral, and this is basically how the coral lives. Okay, so it's an animal that learned to live like a plant. And if we look at the tiniest unit of coral colonies, it's this here. So it's a so-called polyp, but if you turn it upside down, you have a jellyfish. And you all know jellyfish, or most of you, because it's a delicacy here, I learned, okay? So I haven't tried coral polyps yet, but I had them in my mouth while I was diving. And what you can see here, the golden dots, this is basically these algal cells that live inside these polyp tissues. Okay, so what happens is that if the water warms, corals start to bleach. So why is that so, we might actually ask? And I showed you very initially that coral reefs are incredibly rare. And the reason why they are rare is because there's only few places where you can actually grow as a coral, right? You have to be at a shoreline, you have to be at the right depths, you have to have the right light, because you have to house these tiny algal cells to do photosynthesis. So corals compete with each other for this benthic space to grow, and what that leads to in evolutionary terms is that they become each other becomes competitive, so they maximize growth at the summer temperatures. And of course, if you maximize something and you give everything, you become very sensitive to any increase in temperature, which is what we see here. So small long-term increases of temperature actually trigger this process of coral bleaching. Okay, only 1 to 2 degrees Celsius above these summer means. So somehow corals ended up in this evolutionary road that they are become inadvertent victims of climate change. Because of their biology, they had to maximize basically their growth at the temperature, or their optimum at a temperature that is very close to the summer temperature, and because we now have this slight increase, this is actually detrimental to them. So we saw that single corals bleach, right? But then how does it actually look like? Do you have bleaching everywhere? And I was privileged enough to go on this Tara Pacific expedition. So this is the boat. It's 30 meters long, and it was built to swim in the Arctic. So what does that mean? It's made completely out of aluminum, and it floats on the ice, okay? Which is the most horrible thing you can have if you're on the ocean, because it's literally a nutshell where every wave movement goes with the boat. So I was excited to go on the boat, and then I got sick for 5 days. It was really not pleasurable. But I was on a part of the expedition that brings us to the middle of nowhere. Okay, Ducie Island is actually very close where the mutiny of the Bounty happened, if you guys are familiar with this book. It's like 2 days away. But so we sailed for 6 weeks, blue on the floor, blue in the sky, and then this tiny island appears here, Ducie Island. So this is as far as you can get away from any direct human impact, and this was my first dive. This is actually me in the water. So you can see crystal clear water, right? But then everything is bleached. So this was very shocking and kind of a reminder that it's very hard for corals to escape this, and that it's a global phenomenon, coral bleaching. This is then the more, if we look at the numbers of it, this is actually from the World Wildlife Fund report. So this is very important to acknowledge because, of course, in terms of geological times, humans don't become very old, right? So we are kind of guided by the experience in our lifetime, but things change so quickly now in the environment that even within everybody's lifetime, you can already feel the difference. Like in my case, I remember that we had snow in the winter in Germany. We still have snow every now and then, but not like what we had as kids. And it's the same for corals and coral bleaching. So corals bleached before, like there were isolated episodes, but this first global coral bleaching, this first mass coral bleaching actually appeared in the 80s, and then we were 20 years away from the first global mass bleaching. And then you can see what I told you about the tropical storms in Hong Kong, right? So with time moving forward, the events become more frequent and they become more extreme. So the third event, which essentially ran from 2014 to 17, for the data that we had, 80% of the coral bleached and 35% died. So this is the equivalent to, if you would say that in the course of 3 years you lose 35% of the ocean, of the tropical rainforest, if this would happen on land. So this is pretty much as drastic as it gets. And ironically, we just published the paper on the data analysis from this third event while this fourth event actually unfolded. So this is also almost 2 years ago and still ongoing. All this red, yellow, and purplish colors, this is pretty much our ocean. The blue thing is where there is no stress. There's pretty much only one area left where there was no stress, right? But the warming is now so extreme that these people that analyze these data, they have to come up with new scales. So, and this is very dramatic, right? And this is what I'm talking about within lifetime. So you can see that here we have 3 alert levels. 5 years later, we have 5 alert levels. So it becomes so extreme that you have to basically extend the scale of what is happening. And somehow we have to remind ourselves that this is not normal. Okay, and then of course it's me, a scientist, and scientists usually think their topic is super important and I have to warn everybody. But what I'm telling you here is basically, how do you say, well, literally the accepted consensus opinion from governments and stakeholders and politicians worldwide. So the IPCC, the International Panel of Climate Change, okay, this has all these people, scientists, politicians, governance, stakeholders, lobbyists, and so on. They release a consensus document, so this usually has the most benign language, right? Everybody needs to agree what is in there. And in this special document of how does our world look like at 1.5 to 2 degree warming, they literally say that we have a very high risk of losing 70 to 99% of corals. Okay, so this is not an over-dramatization by scientists. This is the consensus opinion globally. So we have to do something about it, basically. And well, yeah, so I don't know how you feel about it, but I run into a lot of students or people generally that find all of this overwhelming, right? Because like working with this, you still have to find your happy place in life. I mean, being depressed all the time doesn't help anyone. But so one thing that helped me in answering this question for me was a meeting in 2018. So this was 2 months after this IPCC report was released. And yeah, you can see me. I'm still young and fresh 6 years ago. Or 8 years ago, and Prince Albert from Monaco. So he's actually an ocean activist. Okay, he's an avid diver. He does a lot for the environment in Europe. This is very well known, and he invited basically influential coral reef biologists to answer this question. Okay, everybody is talking about the reef crisis, but what does it actually take to save reefs? And one thing we did in this exercise is we did a modeling of how high frequency bleaching looks like moving forward under 4 different scenarios. And these 4 different scenarios is basically keep carbon emissions as it is, or lower carbon emissions with 2 other options, adaptations and no adaptations. And these adaptations is a broad term, so it can be societal, socio-economic, and scientific implementations or solutions. And there's various important things which I took home from this. So the most important thing, of course, is that, well, there's only one way out of it, is the most important message, right? You can see that these 3 trajectories, they basically these 3 trajectories, you all end up in the 100% bleaching. So this is where we don't want to be. You want to be down there in the lower right. There's only one trajectory, low emission with adaptation. But what this means is that this carbon emission thing is basically the non-negotiable part. Okay, and I'm saying this because now I run in conferences where they tell me, well, you have engineering solutions, so we can go ahead with carbon emissions. No, we can't not. This is the prerequisite. Somehow we need to find a way to curb this, okay? But the other important part, and this is where I get my hope, it's essential and necessary, but it's not sufficient in its own right to actually make the change, okay? You still need this adaptation element, and this is pretty much what I talk about, how we mitigate this ecological collapse. So I'm not the person, or my job as a coral reef biologist is not how we can curb CO2 emissions, but I can help with this adaptive part to make sure that both things meet. The other important message is, of course, that if we follow this model, and if the model is correct, this needs to be fixed in our lifetime. Right? So it's a responsibility, but it's also a motivation, I think. So we have to do this now. And I looked up this number for you. So what we do is completely insufficient. So who knows what the Apollo mission is? Putting a man on the moon, right? This was the big space battle between the US and Russia back in the days. So back in the days, they invested $25.8 billion, which is nowadays the equivalent, so tenfold, $257 billion, to put a man on the moon, just to achieve this one thing. This is the current spending, and this is under President Barack Obama. So I don't know what this number looks like now, but even under a president of the US that acknowledged the problem, it was 16.5 billion, so 6% of what it was worth to us to put a man on the moon, okay? So there is money to do this, we just not invested into this cause. Okay, sorry about that. But so I already told you this, right? The adaptations is a broad term that incorporates biological, ecological, and socio-economic interventions. And I create out the CO2 mitigations for a reason I already told you. This we need to take care of, but this is not what I'm talking about. if we talk about adaptations in basically, and what I can do as a coral reef biologist, there's pretty much two things at my disposal. It's conservation, which you're all aware, or which you're all familiar with. It's conserving native habitat. But in many places, or in a lot of places, this might actually not be enough. And then there's active intervention, which is a newly coined term, which I actually like, because restoration is already a problem. If you restore to previous state, we already know that it doesn't work, because we see how it works in nature, right? Restoring to previous state or situation is not good enough. So we kind of need to rebuild it. But the broader term is active intervention. So I talk a little bit about both. And so this is, I dove in this reef. This is in Australia, in the Great Barrier Reef. You have to go very far out. But Australia is very special because it's a small country. It's an industrial country. They don't rely on coral reef ecosystem services. They don't need to fish, and so on. So they can actually afford, quote unquote, to leave reefs alone. But this is a normalization how many fish you would see in a reef, okay? If you, and I encourage everybody, so there is a diary of James Cook. Who knows who James Cook is? He's the one that, quote unquote, discovered Australia. So he arrived in Australia, and he has a diary where he describes when they see the first coral reefs. So apparently it was shark-infested waters, and they were jumping around like dolphins. So the coral reefs back in the days must have been completely crazy. I think we cannot possibly imagine, but this is what I remember when I look at this picture, right? So again, this normalization, if I go diving nowadays, I'm very excited to see a few sharks, but there was a situation back in the days where ecosystems like coral reefs could like support immense amounts of organismal diversity. Okay, but back to the topic. So if you have to save, I just told you there are so many species in coral reefs. How do you actually start? But this is the proxy: saves the corals, saves the reefs. Okay, so corals are literally the prerequisite for everything else. If you make sure that the corals are there, the rest will follow, which is important to tell people, because I work in Indonesia a lot, and they care about corals to the extent that this is where they fish. Okay, but you have to tell them that if you save the corals, you save the fish, and they have more fish, but you need to make these connections. Over lunch, I just had a discussion about how human health is connected to ecosystem health, right? And this is one example. And one thing we want to focus on is so-called bright spots, which is also a pitch to ocean optimism, right? I now say this, and maybe I take away a question from somebody, but if you think about it, I would always prefer to live in the future than in the past, because I think we have the technology and we can do something about it, and this bright spots goes into this ocean optimism thing. There is reefs that do much better than expected, and I think those are worthwhile to basically understand what are they doing different from the rest of this world or the rest of the reefs. And Dan Barshes, who was my partner in crime back in the days, we actually had the same idea about 10 years ago. So you then start to look at the data and the published literature, and it turns out we actually don't have such a good idea where these bright spots or these super reefs exist. And of course, it's nice to go diving, but it's also, generally, it's hard to access a coral reef, okay? A, you need to be in the place, B, you need to have diving gear, and then you need to go underwater, and then you need to measure the corals underwater. So you're always 1 degree from convenience, okay? So doing experiments underwater is very complicated. So when we started thinking about this, we wanted to have some standardized coral climate test that we can quickly deploy to measure corals. And then if you do this, you can basically identify corals that are very tolerant, stress tolerant or resilient. And then you can compare to places where they are less stress tolerant and resilient. And it's also important that it was not meant, well, it's like a COVID test, okay? Well, every one of you is familiar with that. You do COVID tests. If the thing lines up, you know that you have to pay attention and that you are in all likelihood to have COVID and so on. And this is a similar idea. It's not that this is the be-all, end-all answer to why these corals are tolerant, but it's a way to basically see if you identify the coral of interest. So this is how this looks like. It's basically 2 boxes with 4 tanks, this 1, 2, 3, 4, and you have these little coral fragments in all of it. You can actually pack it in 2 or 3 suitcases, and you can also deploy it in hotel rooms and in Airbnb. So this is in a very fancy resort in Abu Dhabi. Don't tell the people, they are not happy about this. If they hear it, and you know how it is, if you tell people that you're doing experiments in their hotel room, it doesn't go so well. This is an Airbnb where we did it, but this was a formerly living room. We actually have time capture videos of how we transform the living rooms into the laboratory. But you can do it, that's the whole point. Right now you can actually bring your lab to the reefs, which is much easier than bringing the corals to the lab, unless you live right in front of the corals. So this is now the sciencey part. What we basically do is it's like we put the corals on a hamster wheel and we measure how fast they can run in a very quick succession. And as I said, this is a proxy of which corals do well. Let's see if this video works here. Yes. This is actually how we collect them. So this is an underwater drill. You can see it's very quick, and then you get these little pieces out, and these little pieces is what ultimately ends up in the test. So this is actually not very intrusive for coral. If you do this, you can basically put a little paste over it and the coral fully recovers from it. So funnily enough, there are certain places where we went to sample over and again, and we literally we found our little paste that we put over the coral. So we drilled in the same site. It was completely indistinguishable. Yeah, but what you can see, so these 2 pictures, this is actually 10 pieces from 10 colonies at a control temperature and at a very high temperature. And you can clearly see that the top left one is very white and the top right one stayed dark. So according to our test, that would then be, the right one would be a tolerant coral and the left one would be susceptible coral. Okay, this is, don't know. So if you have 4 temperatures, you can do something that is called a dose response curve. It's actually borrowed from the medical literature. You can see the response with an increase in stress, and you can clearly see that the coral, the red line, is the dose response curve of a population that is tolerant, and they can hold out much longer. If you look at, it takes a lot of temperature for them to lose the performance in comparison to the blue population. But now we basically have a scheme of how we can differentiate this. This is the point to make. And what we found, in essence, as I said, back in the days, I was in the Red Sea. The Red Sea is a very narrow, very deep, and very warm sea. And but it's also a very there's also a very strong gradient, which you actually have here in Hong Kong waters too. So gradients are always cool for science because you basically have the same geography and site, but you have different environmental conditions, right? If I compare corals from the Red Sea and the Great Barrier Reef, arguably it's 2 very different sites, 2 very different evolutionary histories. But if you stay in the same site, you can actually attribute the difference to the environment. So turns out that these northern Red Sea corals are super temperature tolerant, which you can see here. And the reason for this is actually in the Bible. So you're not too familiar with the Bible, me neither. But you probably remember that at one point the people of Israel went through the Red Sea. So the Red Sea dried out. This is a historical fact, okay? And the corals that had to repopulate the Red Sea, they all came through this small opening. So they all had to go through this very warm environment here in the southern Red Sea. So even the corals that ended up in the very north, they were selected through this And this is what basically explains this phenomenon that we measured, right? So we have corals in a much colder water, but because they had to migrate through this very warm zone, they basically kept their evolutionary advantage, so to say. And this is actually nice because if you count the days we were in the water, it was 2 weeks, so very quick. So what comes from this is basically this idea, okay, so now you have a test system to identify this naturally resilient coral, and A, you can do this to inform conservation, so where do you find them? And B, you can do this to inform active intervention approaches. So how are resilient corals different to stressed corals? And this is what we did. Basically, so Paul Allen is a philanthropy. He's actually the co-founder of Microsoft. So we got money to deploy this system globally and measure corals, which is very nice. And what we are currently, here you can see that there's actually a lot of people that do this, which is great, right? Like if a lot of people do the same thing, you have a lot of data to compare with. But what we're building right now, are you using Google Maps here, or is there another app to navigate? Okay, yes. So we are building Google Maps for coral conservation. So all these experiments that we have, we upload to a database, and then you can either select your species of interest or your region. You can identify who did experiments there, on what species, and B, so you have this convenient slider. So in the day of smartphone, each of you could now do a scientific study. Where do you find the most thermally tolerant coral? And then you could basically match this with satellite data, and you could check what are the environmental conditions that led to this. So this is an ongoing effort. This is basically the beta release, but you can play around with it. It works already. I think we have 250 experiments already uploaded, but there's much more to come, which I think is an exciting initiative. And yeah, so having talked about this conservation, this other aspect, active intervention, right? I already explained it. So who has heard the term microbiome before? Gut microbiome, so, okay. So bacteria are actually good news, okay? Bacteria are not always bad. So each of us, we have a lot of bacteria. I think we are actually outnumbered 1 to 10 by bacterial cells. Yeah, we have 10 more bacterial cells on us and in us than human cells, okay? And whatever we eat, we couldn't use if it weren't for the bacteria. But somehow it took a very long time for the scientific literature or scientific approach to consider that, well, there might be a reason why we have so damn many bacteria. So the sum of all bacteria is called the microbiome. Okay, biome is usually the entity of something in the biological world, and the microbiology biome is the microbiome. So as I said, the human microbiome is probably what you are most familiar with. So these studies are, it's not just, how to say, it's not just tuning your car, okay? There is, your mood depends on the bacteria you have in your gut, which in turn, the bacteria you have in your gut depends on what you eat. So this, you are what you eat, has now a scientific correlate for real, okay? So this goes actually very deep. So accordingly, what I told you before, the coral animal and the algae, now we have a third player, which is the bacteria. Back in the days, we did an experiment. I have to see how I'm with time. Back in the days, we did an experiment. So like even in the science world, not everything is ideal, okay? Like if I learned one thing, then that humans are creatures of habit, and it's very hard to break habits, okay? So in the science world, you learn something when you're a student, and then you believe that this is true for the rest of your life, but we have to constantly update each other, and we have to read more papers and do more studies to basically stay up to date. So classically, these algae are associated with temperature tolerance in coral, but in this experiment, we basically showed that also the coral bacteria are important. So conveniently, you can how to say this in a nice way, you can chunk coral colonies into various pieces, and because they are colonial, each piece of this coral colony is a real representative of the colony. So you have the same genotype in different experiments with the same life history, which is very powerful for experimental design. So what we basically showed in this study is that we could transfer microbiomes from heat-tolerant to heat-susceptible corals, and then these corals actually behaved identical to the heat-tolerant corals. So irrespective of host genotype or host DNA, which all stayed the same, so you literally transformed one coral into another coral by means of their bacteria, which of course is pretty good news because bacteria are much easier to manipulate or to engineer than any other entity, which of course led to this idea. So now maybe you can do microbial therapy, which is written here. So use good bacteria, associate them with stress sensitive coral and makes them more resilient, which is actually what we did. There was one experiment where we used a group of good bacteria, we put it on coral and we run them through heat stress, and these corals became actually very tolerant. So to the point where we achieved a 40% increase in survival. So this is absolutely amazing. I really had year-long problems believing this, because in science you usually have very small effects that you are excited about. So having increased the number of surviving corals by almost half, this is pretty impressive. So, and again, coral reef biology and the medical science field is actually not that different, right? So they have an approach which is called microbial transfer therapy. So people that undergo antibiotic treatments as a consequence of, for example, organ transplants, you can imagine that you basically have to bleed the entire, you have to curb the immune system of these people. And what usually comes with that is also an antibiotic therapy. So these people have a completely destroyed microbiome. And it's very hard sometimes for these people to rebuild the good bacteria. So one thing that was actually shown to work well is to isolate bacteria from healthy individuals as donors and to give it to the susceptible individuals. And then you can reconstitute the microbiome. So this is pretty much what we are doing here. And like in humans, it actually works amazingly well on average. But there's two things that are not addressed with this. A, we actually don't know what the characters of these bacteria are. What are you looking for in a bacteria that makes it a good bacteria? And B, of course, whatever you put on there, you want it to stay on there. So this is the two kind of things that you have to address, the two bottlenecks, and this is actually very well acknowledged. So believe it or not, or I don't know if you know it, so this microbiome engineering is actually very broadly used in the agricultural world, which you can imagine, right? Crops, they need to grow well, and you usually supplement them with bacteria to make them grow better and so on, and they all have this, or there's a lot of studies that acknowledge this so-called boom and bust. So you give this good bacteria, you have a good effect initially, and then the effect wears off. What is the problem if it wears off? You have to reapply it. What is the problem with reapplying? Well, in the coral world, you have to go diving, and then you have to put it on every coral. And I can tell you, I think there's an average of 10 million coral in one reef, if it's a good reef. So it's impossible, pretty much. If you would take all the coral reef biologists in this world that dive, even for one reef, you couldn't achieve it, probably. So you need to find another way, which is where Professor Hai-Wei Lu comes into play. So basically the way you do it is you look for these traits in our language that you assume have a beneficial effect, but you don't really know if they have an effect. But what Hai-Wei did, he inverted the search pattern. So the other side is evolutionary. If a bacteria commits to a host, there's very clear genomic signatures which you can find, and you probably all heard that sequencing became very cheap. right? It's funny, so 20 years ago, the human genome was sequenced, and I think, I forgot, it was a lot of money. So we are now really in the age where I could basically swap my saliva, I could extract DNA, and I could sequence my entire genome for 100 Hong Kong dollar or US dollar? 100 Hong Kong dollar, very cheap. Okay, well, you still need somebody to analyze it, which is much more expensive, but the data you could generate for 100 Hong Kong dollar. So this is not the bottleneck is not the sequencing anymore. So this is just how we look at stuff. But there's basically a lot of bacterial isolates that you can compare, and then you find some of these isolates that have this signature of undergoing host commitment. One is called MC10B4, and this is a great example how science doesn't come up with names that are very accessible to the public, right? MC10B4 is very boring and unexciting if you put this in a news headline, but it's actually how we talk to it, so we have to come up with a better name at one point. But so this bacteria, it basically, there's a lot of things on this slide. I know I'm trying. So we put it on corals in the field, and this is the Hong Kong summers before, and this is the Hong Kong summer 2 years ago. So this is off the charts water temperature. Okay, this is what we just talked about. But in this summer, and we don't know how the next summer looks like, this is when this experiment was done. So you applied this good bacteria to the corals in the field, and these corals actually did better in a natural reef, in a natural environment, in a natural heat stress. So this is as close as you get to this proof of principle. And they also stayed associated over this entire time. So they had a beneficial effect, and they were still present after 8 months. So really, I'm very excited about this. You're very excited about this. Because now you have something that is, you can actually scale, and you address these two things, right? Like the long-term association and host benefit. So we received funding for this international collaboration to actually do this in a field site in Indonesia, which is important for several reasons. You could do it here, but doing it there actually also proves the point that you can do this anywhere in the world, right? Because this is not arguably a super high-tech field site, right? But I'm pretty confident we will make this work. But this is actually, we are building a field lab now. This is a 3D rendering, and tomorrow I'm flying there, and then we can compare how it looks in reality. So this is where we do this. Gili Asahan is a very small island. This is this local community that we work with. But the point is that sometimes I feel like Homer Simpson. There is this retirement home in The Simpsons, and they have this sign on the door, "Thank you for not discussing the outside world." Right, so I'm now in the opposite. I'm discussing the outside world with you because it's important, because as a scientist in coral reef biology, it's not good enough to do the biology. You actually have to go out and talk to people and try to make a difference, which we are trying to make here. And also because it's now this case that I literally, I'm long enough in the coral business to actually see how bad things become over a relatively short time frame. Okay, and with that, I want to finish. So the bottom line, basically, the situation is dire, but we can and we have to do something about it, and we can and we have to do it now. And I actually, in my week here, so I will definitely be back, but there's also a lot of willingness to collaborate, and there's a lot of understanding that we have to tackle these challenges together. And I think this is really, we are all world citizens, right? This is how we have to move and think about these things. So with that, I thank you for your attention. Thank you.
Thank you, Professor Voolstra 同我哋分享氣候變遷對珊瑚礁嘅影響。而家進入問答環節，請 Professor Voolstra 同埋羅教授對台前就座主持問答環節。 May I now invite Professor Law to the stage again to take a seat and moderate the Q&A sessions with Professor Voolstra. Professor Law, please. 歡迎現場觀眾舉手發問，而收看網上直播嘅觀眾呢，亦都可以留言發問嘅。
So we start from the.
Thanks, Chris. Hi. That's a very amazing talk of how corals can be important in the future. I'm very interested to know that based on your example studying Red Sea, how coral evolved northward. And based on this example, my question is, where on Earth do we find this good bacteria, like geographically or in different depths or in different habitats? Is it like places in Hong Kong or in like in what scenario can we find this good bacteria so we can go there and then sort that out?
Yeah, that's a very good question and exactly what we're trying to figure out. So hopefully good bacteria are everywhere. So I didn't mention this, but it's not that. So if you go diving in a reef, right, you actually, in a bleaching event, a lot of corals are white, but some are dark. And these dark corals, this is what you have to go for. So there is a big difference. It's like if you take all the people from Hong Kong, right? The average size is one thing, but then there's smaller people, and then there's taller people, and then there's very tall people, and there's very few very tall people. But this is the corals you have to go for. So ideally, either we go into every region and we look for this very great coral in these regions, but of course, the best thing would be that you find this one or many bacteria that can help every coral, and that that is basically without boundaries. But this is active research, and of course it comes with concerns about ecological impact and so on. So you have to very carefully navigate this, right, how you do this, because everything we're doing right now, if you use local bacteria, you're only bringing something back that was already there. If you would take a bacteria from one place and put it to another, that's a different thing. Also, arguably, humans are the best vectors. So we constantly move bacteria around in the world. When I take a flight from Germany to here, I bring all of my bacteria and I touch door handles here and various things. So like we have to keep this in mind, that organisms are actually pretty well equipped to like keep bacteria at bay, because we're constantly exposed, right? I personally, like, I think it's worthwhile trying to understand this, and if we would genetically modify these bacteria, this would be another story. But like, bringing bacteria into foreign environments happens all the time, and we're not changing ecosystems all the time. This happened for the last 500 years that we move stuff around.
Question?
Thank you for the talk. In the talk, you mentioned that when the heat is going up, the coral are going to be extinct or to be whitened. However, in my knowledge, that is like today is the ice age of Earth, and as coral reef can survive all those more extreme environment in Earth history, then why this little warming can cause such a disaster in today's Earth? That's my question. Thank you.
Yeah, it's a good question. The problem is the amount of change per time, right? So usually things happen on a much slower time scale. But what we have now is that we have doubling warming rates every 10 years, which is way quicker than in ice ages. And it's simply not enough time for organisms to evolutionarily respond to it.
Once again, thank you very much for the lecture, Dankeschön. And my question is like, because we mentioned like the evolutionary mechanisms, right? So does it mean that if global warming stabilizes in the long run, let's say we stop emitting more carbon and like global temperature stays the same, like over time would coral reefs actually adapt to like the changing temperature, or like is it possible for them to go through the whole like natural selection and then become a little bit more heat resistant than they were, say, like 100 years ago? Thank you.
Yeah, good question. It's already happening. Like corals on average are 0.5 degrees Celsius more temperature tolerant than 20 years ago. But there's two ways of how you can achieve this, right? You can kill everybody that is below that threshold, or you can adapt to have more of that threshold. So what we are seeing is that actually the corals that cannot make it die, right? So the unit of evolution is population. If you have one individual, it's not, you cannot evolve anything because you select individuals within a population. So the problem we're doing right now is we're like reducing genetic variation drastically. But I haven't answered your question. If there was a lot of genetic variation left, which is a big if, but corals would eventually adapt to it. But the problem is that adaptation can also work most efficiently under consistent conditions. What we're doing right now is that environment is constantly changing, and you cannot adapt to a constant change because it becomes a runaway game. This is actually one of the biggest problems. It's not that organisms cannot adapt, it's that they cannot adapt under non-constant environments. And I think this is a lot of people don't acknowledge this. So thank you for saying this. But again, once we're at the point where we stabilize, there's only very little genetic variation left for selection to act upon. So it's a convoluted problem.
More question? Yes,
Christian, thank you once again for the talk today. And this is the second time that I saw your picture, you diving in the middle of nowhere ocean, where all the coral were like bleached. And my question first is, do you see any other life form in that part of the ocean? Yeah, yes. So then my question is, so from your talks, it seems like to me, coral is the origin of everything. You have coral, then you have a very good ecosystem. You can support different life forms, and everybody's happy, and sharks are swimming and everything. But once the coral start to bleach, then I got a message that everything would just stop. Everything stopped, and then things start to deteriorate. And in the end, the only thing that we can do is to restore the coral, make them live again, and then everything come back. So my question to you actually is, so if I'm the other life form in the coral reef, then when I see the coral start to bleach, then what should I do to survive, instead of just seeing the coral start to bleach and I can do nothing? Any other just like a food web, other than like eating on coral and rely on coral, what else can we rely on as other life forms in coral reef? Thank you.
Yeah. So first of all, you're a very good observer because this reef, it's very wide to illustrate this fact, but there's also zero fish. And there's not zero fish because the reef is bleached. There's zero fish because it's so far out, right? So generally, there's not so much biomass. So in that sense, it's not representative of the typical reef, I would say. But so if you would put a coral in a mixer and then sequence it, you would probably find 1,000 species associated with it. So the coral itself really is the habitat for a lot of species. The third thing is that if they bleach, it's still okay, but the end state of bleaching is that the coral basically loses the nutrition, the algae, right, the source of nutrition, and then over time it basically dies. And once it dies, it looks like a rock, and it dissolves. And then, of course, the habitat for these organisms is destroyed. I think what you're getting at is artificial reef structures. So is that what you're aiming at? There is basically attempts to recreate, right? So a coral is, of course, great, but the habitat aspect of a coral is that it creates this like complex little structure where organisms can live on. So that function can be partially substituted by artificial structures, which people actually try to do, right? But you always lose a certain aspect. So I think these are good intermediate solutions, but they are not end games. It's not an end state. It's not sustainable, right? At the end of the day, there's so many interactions that we are not aware of, and we know that the biocomplexity of these artificial structures is 10 to 100 times lower than on an alive coral. So they can substitute some of the function, but not all of it. And we don't exactly know why or what is missing.
Another question?
Thank you very much for the talk today. I have a question trying to correlate the health of the coral system in the ocean to the how will I say, to the appearance or disappearance of fish in the ocean. Normally, if all other conditions remain the same, the fish will go for where the food is. And that's basically, they're looking for zooplanktons or the microorganisms. Would you agree that if we have a healthy system of coral in the ocean, that's where we would expect to find a good population of the food for the fish to begin with? Okay, now we are getting in your presentation, basically all over the world, we have problems with the coral system. So that means the food available for the fish are decreasing further and further. Is there a way to correlate the health of the coral system to the availability of the fish that we can find, for human, for human consumption? Is there a way to correlate that?
Yeah, there's a very direct way. So just, it's all very good what you're saying. The relevance of coral reefs is not always adult fish that we want to catch, but larval phases of fish are usually tied to coral reefs, right? Because if you're young fish, you need to hide from your predators. So this usually happens in the coral reef. So even this, what we call pelagic species like tuna and the stuff that is in the open ocean, they have a life stage that is in the coral reef. So you still need corals even for many of those fish species. There is direct research, so fish can actually smell healthy reefs. And one of the molecules is DMSP. So it doesn't matter what it is. It's a sulfide compound. But so you have to think in terms of biology and evolution. It's so fine and so important that larvae can smell where there is a good reef, which is where they spend their time. So if the reef is bad, these larvae don't go there. So there's really a direct biological mechanism where you have less fish in a deteriorated reef because it doesn't smell the part that they want. There's actually now research where you replicate this smell with chemicals to attract the fish. So this is how this connects. And then what else did I want to say? Yeah. So we do restoration projects, and as I said, that it's really like this foundation species, that corals is the foundation species of reefs. This cannot be overemphasized. Like if you plant one coral on a rock in terms of restoration, and you come 2 weeks later, there's a lot of other organisms that come with this piece of coral. So this is part of the research that we're doing, right? Because as I said earlier, for a lot of people, the coral are not so important, which I can even relate to. But if you show them how many species you restore. How many species do you restore when you plant one coral? If you show them that there's hundreds of other species that follow, then I think this is very impressive to a lot of people, and then they understand the significance of it, right? It's like you plant a tree and then you have birds that sit on the tree. You're not restoring the birds, but you're restoring the tree, and then you create habitat for other species to follow. Does it answer your question? Yes.
You mentioned that the microorganisms are essentially food for the larvae or the baby stage of the fish. Okay, for sure, that's true. But is there any correlation between the health of the coral system to the adult fish population? Because nowadays, when we see that the marine resources all over the world, I would say erratic, completely erratic.
Eradicated, yeah.
Yeah, erratic. Okay, there used to be seasons. Okay, for this species in this part of the ocean, you would normally see them. They will come, let's say, from May to October. You don't see that anymore.
Yeah.
All over the world, and it's not only for one species. Essentially, it's happening for all the species, and it's driving the prices of fish or any marine seafood up the roof. Fishermen don't know what to do. They don't know when they could go out. You have good fishing for 1 week, and all of a sudden, all the fish are on vacation.
Yeah.
So what are we going to do about that? Is there a way to correlate this with the coral reef health population?
Yeah, okay, there's two things that you have to do. This one thing I actually didn't mention, right? So if there's no coral, there's no fish, but this is the one side of the problem. The other problem is that we catch a lot of fish, right? Which is what you're alluding to. So like what I answered so far is a way to basically replenish the potential for the next generation. But what we actually do is, if we don't find fish anymore, we go further out and we find other places. And so we're literally going down the population size of these fishes. And there's actually studies like these bright spots also exist in the fish world, where there is places where you have a lot of fish, more fish than you would expect. And then they look at what happens there. There's actually people or tribes, indigenous people, that somehow learn you cannot fish everywhere. That is the bottom line of my answer. Okay, you have to leave populations alone in certain spaces. So these indigenous people, it's actually entrained in their culture, and so that they have seasons or times where they go to different places. So they always have certain sites where population recover while they fish from other populations. And I think what we are doing right now is we fish a population out and then we go to the next population. So in order to make this sustainable, we basically have to leave some populations time to recover. And this is irrespective of coral reefs, I think. So to answer your direct question, we cannot measure this correlation because we also keep fishing away the fish, right? Otherwise, I'm convinced you would see that correlation. Yeah.
So we have one question from Zoom. So let me read out. So obviously, one of the issues, not the major issue, are the hydrological water flows governing a reef. The amount of water and the nutrients define the biogeochemistry of coral reefs. So basin scale, basin-wide management is essential, which would also include compound of emerging concerns. Probiotics can be good, but it is a very localized and volatile intervention that must be coupled to large scale ecosystem management, right?
Yes. So yes, good question, because it let me emphasize this. So solutions are multifactorial, right? There is no silver bullet, but there's hopefully many silver bullets which together make a difference. So, which is kind of what I tried. That there's instances where we have to conserve. There's instances where we have to actively intervene. But there's certainly a toolbox of interventions where we have to understand A, how does this toolbox look like, and B, which of these interventions are we using together at that particular reef. So yes, I certainly different solutions which tackle the different origins is definitely important. And also in this active intervention world, we actually have very little understanding about this, right? We don't know if you tackle two problems at the same, if you basically get a linear increase in the positive effect, or if you have this, what we call synergistic effects in the world, right? That two good things are better than the sum of the two things alone, but they make a much bigger difference. So I think this, we really have to put more research into what is the interventions that make the most difference and how do they are best deployed in parallel or co-occurring.
Another question, yeah.
Hello, thank you for your amazing speech. I have two main questions about microbia. So I have a question like, would you and your team choose specific strains of a bacteria introduced in the coral reef? Because as we all know, as the same bacteria in different strains, they have different function, like immune system, digestive system, like that. So yeah, this is the first question. The second question is, do you and your team have any follow-up plans? Because we know that if we introduce a new microbiome and a coral, maybe there will have some environmental change and some bacteria will enhance or increase the overpopulation that quick and will affect the whole ecosystem of coral. So this is my two questions. Thank you so much. Thank you.
Yeah. Sorry, I thought about the answer for the second question. Was so specific? Yes, yes, very, very, very specific bacteria. Right. I didn't talk about. I skipped over this part, but finding this one bacteria, this is one strain of one population of one species that actually has this effect. So, but again, this is like now very sciencey, right? This is, we don't actually, well, we conceptually understand what we have to look at, because it's exactly what you say, right? There's a lot of difference within species that is not acknowledged very well, especially in ecology. We have, conceptually we know this, but how we tackle this in scale is a big question. So for this study, 400, 800 isolates.
For their study is 400 plus.
Yeah, so 419 isolates to find this one strain, right? And one strain is one genotype in one population of one species, as I said, so you're spot on. Second, this is, yeah, very convoluted. Like, of course, you have to acknowledge that there is some risk, how real this is. We have to see. I don't want to say that there's no risk, but A, there's also risk of doing nothing. B, we're constantly changing the environment, even though we're not doing it in terms of a scientific experiment, right? But if you go in the ocean nowadays, I mean, so I did, I was in the submarine at whatever 4,000 meter steps, and the first thing I saw was a garden chair, a plastic garden chair. Like, this ecosystem has nothing to do with humans, but the first thing you find is a plastic garden chair, right? So you could argue that the order of magnitude of how we change the system experimentally when we actually know what we're doing is potentially smaller than other things what we do. But of course, I grant you that the bacteria we use are potent, because that's why we use them. But again, yeah, I think we have to do the science and we have to figure it out. And this is really important. And this is, of course, detail. But like we're not genetically modifying these things, right? The way we're doing it currently, you find these things in their native environment and you put them back. So arguably you increase the abundance of something, but you're not changing the genetic material of something, right? Because that is really a concern in a lot of these things, that you introduce foreign genetic material that then goes haywire. So the genetic material that is in this system evolved to be in harmony with that system, and this is not what we are tinkering with. And this is also kind of where my boundary would be, I think. So it's personally, it's also at a point where I can still justify it, right? I'm not the guy that, oh, because we can do it, we have to do it, and then we deal with the consequences later.
Very good question. Other question? Great opportunity. Change ideas with the leading scientists.
Oh, yeah. Great question.
So if we want to participate in the project that you talk about in Hong Kong, so how can we start? I mean, Professor Law, maybe you can give us some opportunities or let us know how can we contribute to Christians and your research.
First, I was saying, of course,
You can answer that. Yes. You should answer that. So no, because I was smiling. Like in Germany, you are not allowed to receive money from the public as a research professor.
Oh.
No, I'm just saying because I asked, because I run into a lot of people that want to support it also financially, right? And then I'm actually not allowed to do this because I need to stay impartial. And then I told my university, but crazy. So this is kind of what we are currently trying to figure out. So that was my first thought, but now you.
Yeah, so many people actually can contribute, because especially thinking about, because we can involve citizen science project program, something like that, because we need to inoculate under the water, inoculate our probiotics on the corals. So this is also still so we will be doing in the nursery. It's not like in the wild coral nursery. Then we outplant the inoculated coral to the wild reef, right? During this inoculation, we can involve many people who can dive. So they can be trained before they dive in inoculation. So they will be doing properly. So will be a lot of opportunities actually.
Yes, if I may, but this was good answer. But I also wanted to extend this, right? What everybody can do is, I think, I don't know how it is here because you all seem very educated and fans of science and so on. Well, everybody in this room anyways, but science has a very bad reputation at the moment globally, right? For whatever reason, but you can support the scientific method, right? Inform yourself factually and contribute to the societal discussion, because it's not how it was back in the days that the scientific method is basically free from doubt. There's a lot of people that doubt that the scientific method will provide the answers. Personally, I think it's the only way that provides the answers. There is no future without science, if you ask me. But I think it's all we are the society, right? We can push, we want more societal facts going into policy decisions. There's very little hard scientific facts that shape the actual policy decisions if you look at it, right? Because there's a lot of other incentives that need to be considered, but this is where we can all have a part in this. I think the next time you can say, well, let the scientists talk. Don't let the politician talk, let the scientists talk first. And then we go from there.
Okay, so if we don't have further questions, so let's thank you, Professor Voolstra again.
Thank you.
Thank you, Professor Voolstra and Professor Law again. 等我哋以熱烈嘅掌聲感謝 Professor Voolstra 精彩嘅分享。今日嘅講座到此結束，多謝各位現場及網上觀眾嘅參與，期待來年再次見到各位，再見。